Welcome to Inside the Cell, Unraveling Insulin Resistance, a groundbreaking series where we delve deep into the microscopic world of our cells to uncover the crucial role insulin plays in our body's metabolism and what happens when this process goes awry. Insulin resistance is more than just a precursor to diabetes. It's a multifaceted disruption that affects our cellular machinery in profound ways. This series will guide you through a journey inside the cell, revealing how each component interacts with insulin and how these interactions can lead to widespread metabolic diseases when they malfunction. Why should we care about insulin resistance? Because it's at the heart of not only diabetes, but also plays a significant role in other health conditions, including heart disease, obesity, and even cognitive impairments. Each chapter of our series will focus on a different cellular element, from the plasma membrane to the nucleus, to illustrate the intricate dance between insulin and the cell, and how disturbances in this dance can lead to serious health issues. We'll start at the plasma membrane, where insulin first knocks on the door of the cell, asking it to let glucose in. As we move through the cytoplasm and into the mitochondria, we'll see how energy is produced and what happens when this process gets disrupted. Lipid metabolism, protein synthesis, and inflammatory pathways will be our stops as we explore how they contribute to and are impacted by insulin resistance. We'll even venture into the nucleus to see how insulin resistance can change the very blueprint of our cells by altering gene expression. And it's not all about problems. Each chapter will also highlight cutting-edge research and practical steps we can all take to improve insulin sensitivity and protect our cellular health. From diet and exercise to the latest in medical research, we'll provide you actionable advice to help you understand and manage your metabolic health better. So join us as we embark on this cellular journey where science meets everyday health. Stay tuned for an enlightening exploration that will change the way you think about your body's cells and the hormones that help them thrive. Whether you're a student, a health enthusiast, or someone looking to understand more about how your body works, this series will provide you with the insights and tools you need to take control of your health. Welcome to the first part of Inside the Cell, Unraveling Insulin Resistance. Today, we dive into the fascinating world of insulin, a key hormone in our body that plays a vital role in managing energy and glucose levels in the blood. Understanding how insulin works not only sheds light on our body's intricate metabolic processes, but also sets the stage for comprehending the complexities of insulin resistance. Insulin is produced by the beta cells in the pancreas and is released into the bloodstream in response to rising glucose levels, typically after a meal. Its primary function is to manage these glucose levels by facilitating their uptake into cells, where glucose is then converted into energy. This process is crucial for maintaining the body's overall energy balance and ensuring that our organs and muscles function effectively. The journey of glucose from the bloodstream into the cell begins when insulin binds the specific receptor on the cell membrane. This receptor is part of a complex mechanism that acts like a lock and key. Insulin, the key, binds to the insulin receptor, the lock, triggering a series of signals inside the cell known as the insulin signaling pathway. This pathway involves multiple proteins and enzymes and is crucial for the next step in glucose metabolism, the translocation of glucose transporters to the cell surface. These transporters, primarily GLUT4 in muscle and adipose cells, are the gatekeepers that allow glucose to enter the cell. Under normal circumstances, the binding of insulin to its receptor causes the GLUT4 transporter to move to the cell's surface, increasing the cell's capacity to absorb glucose from the blood. This not only lowers blood glucose levels, but also supplies the cells with the fuel it needs to produce energy. However, when insulin resistance occurs, this process is disrupted. The receptors on the cell membrane do not respond to insulin effectively, and as a result, glucose transporters are not mobilized to the cell surface efficiently. This leads to higher levels of glucose remaining in the bloodstream, a hallmark of insulin resistance and a precursor to various metabolic disorders, including type 2 diabetes. In this part, we explored the essential role of insulin in regulating blood glucose levels and how it facilitates the entry of glucose into the cells for energy production. Understanding the foundational aspects of cellular metabolism is key to unraveling the subsequent stages of insulin resistance that we will discuss in future parts. Stay tuned as we continue our journey deeper into the cell, examining how disruptions in this process contribute to insulin resistance and what we can do to mitigate these effects. In the second installment of Inside the Cell, Unraveling Insulin Resistance, we focus on the insulin receptor and the vital process of signal transduction, which is crucial for understanding how insulin helps our cells absorb glucose. This segment explores the intricate dance between insulin and its receptor, a dynamic that is foundational to our cellular health. The insulin receptor is a protein embedded in the cell membrane, serving as the critical entry point for insulin's actions. When insulin, circulating in the bloodstream, encounters a cell, it binds to this receptor. This binding is akin to a key turning in a lock, initiating a cascade of biochemical events inside the cell, a process known as signal transduction. Signal transduction begins when the insulin receptor activates and undergoes a change in its structure, which enables it to add phosphate groups to itself and other proteins in a process called phosphorylation. This activation serves as a signal relay, propagating the message through a series of protein interactions within the cell. One of the key pathways activated is the PI3K-AKT pathway, which plays a pivotal role in multiple cellular functions, including the movement of glucose transporters to the cell membrane. The effective transmission of this signal inside the cell is essential for the proper functioning of glucose transporters that regulate the entry of glucose into the cell. These transporters respond directly to the signals sent by the activated insulin receptor, thus facilitating the rapid uptake of glucose from the blood into the cell, where it is used for energy or stored for future use. However, in the state of the insulin resistance, this signaling pathway encounters interference. The receptors may not phosphorylate effectively, or the subsequent signaling proteins might not propagate the signal as efficiently. This breakdown in communication means that despite the presence of insulin, the message to absorb glucose is not relayed effectively, leading to elevated blood glucose levels. Understanding the role of the insulin receptor and the complexities of signal transduction is crucial. It not only illuminates one of the primary sites where insulin resistance can take hold, but also sets the stage for exploring how these disruptions can be addressed to restore healthy cellular function. As we delve deeper into the impact of compromised signaling in our next episodes, we'll uncover more about the cellular dysfunctions that contribute to metabolic diseases and the potential interventions to mitigate these effects. In part 3 of Inside the Cell, Unraveling Insulin Resistance, we delve into the crucial role of glucose transporters and their function in glucose uptake. This process is essential for providing energy to cells and is directly influenced by the efficiency of insulin signaling discussed in our previous segments. Glucose transporters are proteins located in the cell membrane that facilitate the transport of glucose from the bloodstream into the cell's interior. Among these, GLUT4 is particularly significant in muscles and adipose tissue, where it is directly responsive to insulin. Normally, GLUT4 is stored inside the cell in vesicles and is swiftly mobilized to the cell surface upon insulin stimulation. This translocation is critical. It increases the cell's capacity to absorb glucose, thereby reducing blood glucose levels and supplying the cell with fuel for energy. However, in the condition of insulin resistance, the mechanism of GLUT4 translocation is impaired. Despite the presence of insulin, the signal to mobilize GLUT4 to the cell membrane is disruptive. This dysfunction can be attributed to flaws in the insulin signaling pathways we previously explored, such as deficiencies in receptor phosphorylation or downstream signaling activity. As a result, glucose remains in the bloodstream, leading to hyperglycemia, a hallmark of diabetes and other metabolic syndromes. The dysfunction of glucose transporters, like GLUT4, underscores the complex interplay between insulin signaling and glucose uptake. Effective management and treatment of insulin resistance often focus on enhancing the activity of GLUT4 and other similar transporters to improve glucose uptake and mitigate hyperglycemia. This can involve pharmacological interventions that mimic insulin's activity or enhance its signaling efficiency, as well as lifestyle modifications like exercise, which naturally stimulate GLUT4 translocation to the cell membrane. Understanding the critical role of glucose transporters in cellular glucose management provides valuable insights into how cells handle energy. It also highlights potential therapeutic targets for overcoming insulin resistance. As we continue our series, we'll explore further how cellular energy production is affected when glucose uptake is compromised and how this impacts overall cell health and function. Welcome back to Inside the Cell, Unraveling Insulin Resistance. In this part, we turn our focus to the mitochondria, often referred to as the powerhouses of the cell. These organelles play a critical role in converting glucose into usable energy, a process fundamentally influenced by insulin and directly impacted by insulin resistance. Mitochondria are responsible for aerobic respiration and energy production in the form of ATP, also known as adenosine triphosphate, using glucose and other substrates delivered to them by cellular mechanisms governed by insulin. Under normal conditions, when insulin sensitivity is high, glucose efficiently enters the cell, fueling the mitochondria to produce ATP through glycolysis and the citric acid cycle. However, in the state of insulin resistance, as glucose uptake into the cells becomes impaired, the mitochondria face a reduced supply of glucose. This shortfall can lead to decreased ATP production, affecting the cell's overall energy and health. The lack of energy can further exacerbate insulin resistance, creating a vicious cycle where impaired mitochondrial function leads to even poorer insulin sensitivity. Moreover, the inefficient functioning of mitochondria often results in increased production of reactive oxygen species, which can cause oxidative stress, damaging cellular structures including DNA, proteins, and lipids. This oxidative stress not only contributes to the worsening of insulin resistance, but is also linked to various chronic diseases, including type 2 diabetes, heart diseases, and neurological disorders. Strategies to enhance mitochondrial function are therefore critical in managing insulin resistance. These can include lifestyle changes, such as regular physical activity, which has been shown to improve mitochondrial density and efficiency, and dietary adjustments that support mitochondrial health. Some pharmacological approaches aim to improve mitochondrial function or mimic the effects of exercise, offering potential therapeutic avenues for those suffering from severe insulin resistance. Understanding the role of mitochondria in energy production provides a crucial insight into the cellular level and the impacts of insulin resistance, and underscores the importance of maintaining mitochondrial health for overall metabolic balance. As we continue to explore the effects of insulin resistance on cellular function, the interconnectedness of these cellular systems becomes even more apparent. Continuing our journey into the cell, in part 5 of Inside the Cell, Unraveling Insulin Resistance, we're going to explore the intricate relationship between lipid metabolism and insulin resistance. Lipids, or fats, play numerous vital roles in the body, including energy storage, hormone production, and forming cell membranes. However, their metabolism is closely tied to insulin's function, and disruptions in this relationship can exacerbate insulin resistance. Insulin regulates lipid metabolism by controlling the synthesis, breakdown, and storage of fats. In healthy individuals, insulin promotes the storage of excess fats in adipocytes, preventing lipid overflow into other tissues. It also inhibits the breakdown of fats, which helps to maintain a steady level of circulating lipids and prevents excessive fatty acid availability. However, in the context of insulin resistance, this regulation fails. Cells, particularly in the liver and muscles, become less responsive to insulin, leading to reduced uptake of glucose and an increased reliance on lipids for energy. This shift increases the breakdown of fats, resulting in elevated levels of free fatty acids in the blood. These free fatty acids can accumulate in non-adipose tissues, a condition known as ectopic fat deposition, which further impairs insulin signaling and exacerbates insulin resistance. Furthermore, the excessive fatty acids enhance the production of triglycerides and very low-density lipoprotein in the liver, which contributes to dyslipidemia, a major risk factor for cardiovascular diseases. The interaction between impaired glucose metabolism and disrupted lipid homeostasis creates a deleterious cycle that not only promotes the development of metabolic syndrome, but also increases the risk of developing type 2 diabetes. Addressing disruptions in lipid metabolism is therefore crucial for managing insulin resistance. Dietary strategies that reduce the intake of saturated fats, trans fats, and combines this with increased consumption of omega-3 fatty acids can help improve lipid profiles and insulin sensitivity. Physical activity also plays a significant role as it helps to increase lipid utilization for energy and reduces ectopic fat deposition. Understanding how lipid metabolism interacts with insulin function allows us to see the broader impact of metabolic disorders and underscores the importance of a holistic approach to managing insulin resistance. As we continue with our series, we'll delve deeper into how these metabolic disturbances trigger systemic inflammation and cellular stress. Moving on into part 6 now, we delve into the complex interactions between inflammatory pathways and cellular stress, and how these factors contribute to the development and exacerbation of insulin resistance. Chronic, low-grade inflammation is a hallmark of insulin resistance. This inflammation isn't the type you see with acute infections or injuries. Rather, it's a persistent, low-level physiological response that can disrupt normal cellular functions. In the context of insulin resistance, inflammation is both a cause and a consequence. It can initiate insulin resistance in healthy cells and exacerbate it in already resistant cells. One of the primary sources of this inflammation is the increased levels of fatty acids in the infiltration of immune cells into tissues such as adipose cells. These immune cells, particularly macrophages, secrete pro-inflammatory cytokines like TNF-alpha and IL-6, which interfere with insulin signaling pathways. This interference primarily occurs through the activation of serine kinases, which inhibit key proteins in the insulin signaling cascade, further impairing glucose uptake by cells. Additionally, the stressed cells themselves can exacerbate the situation, as cellular energy production becomes inefficient due in part to impaired mitochondrial function and discussed in our previous parts. Cells begin to generate reactive oxygen species. These oxygen species lead to oxidative stress, which damages cellular components such as DNA, proteins, and lipids, and further promotes inflammatory responses. The presence of both oxidative and inflammatory stress creates a vicious cycle where inflammation leads to more insulin resistance, and insulin resistance in turn promotes more inflammation and oxidative stress. This cycle is particularly detrimental in metabolic tissues like the liver and muscle, where it can lead to more severe metabolic disorders beyond insulin resistance, including type 2 diabetes and non-alcoholic fatty liver disease. Breaking this cycle is critical for restoring normal insulin function and cellular health. Strategies to reduce inflammation and oxidative stress include dietary antioxidants, regular physical activity, and medications that target specific inflammatory pathways. These interventions can help mitigate the inflammatory responses and improve insulin sensitivity. Understanding the role of inflammation and oxidative stress in insulin resistance opens up new avenues for treatments and preventions. As we continue to explore the cellular impacts of insulin resistance, the interplay of these two pathways highlights the complexity of this metabolic condition and the importance of a multifaceted approach to management and treatment. Welcome to part 7 of our journey inside the cell to understand the cellular effects of insulin resistance. In part 7, we're going to explore the impact of insulin resistance on protein synthesis and how disruptions in insulin signaling affect cellular growth and repair mechanisms. Insulin plays a crucial role in protein synthesis, promoting the growth and repair of tissues by activating pathways that lead to the translation of specific proteins. One of the key pathways influenced by insulin is the mTOR pathway, which is central to cell growth and protein synthesis. When insulin binds to its receptor, it activates the PI3K AKT signaling pathway, which in turn stimulates mTOR activity. This stimulation enhances the cell's ability to synthesize proteins, which are necessary for tissue growth and repair. However, in the state of insulin resistance, this process is hindered. The impaired insulin signaling results in reduced activity of the PI3K AKT pathway and subsequently the mTOR pathway. This reduction leads to a decrease in protein synthesis, which can affect muscle growth and maintenance, as well as the repair of damaged tissue. For individuals with insulin resistance, this means slower recovery from injuries, decreased muscle mass, and overall poorer health of tissues. Moreover, insulin resistance can lead to alterations in the expression of various genes involved in metabolism and cell cycle regulation. This altered gene expression can result in the production of proteins that are not optimally functional or can further contribute to cellular dysfunction. For example, in the liver, impaired insulin signaling can increase the production of enzymes that lead to excessive glucose production, exacerbating high blood sugar levels. The consequences of disrupted protein synthesis are significant, impacting not just individual cell health, but also the overall metabolic health of the body. To combat these effects, therapeutic strategies often involve lifestyle changes that can improve insulin sensitivity, such as exercise, which directly stimulates muscle protein synthesis independently of insulin. Nutritional interventions, particularly adequate protein intake and the timing of meals, can also play a crucial role in supporting optimal protein synthesis. Understanding the intricate relationship between insulin signaling and protein synthesis illuminates another layer of complex metabolic networks affected by insulin resistance. So as we continue our journey through the cellular landscape affected by this condition, the importance of maintaining a healthy insulin signaling pathway becomes ever clearer, but not only for metabolic health, but also for the structural and functional integrity of the body's tissues.
Welcome to Part 8 of Inside the Cell, Unraveling Insulin Resistance, where we're going to delve into the nucleus of the cell to explore how insulin resistance affects DNA and gene regulation. This part highlights the critical intersection of insulin signaling and genetic expression, showcasing how disturbances in insulin pathways can lead to significant changes in cellular behavior and function. The nucleus is the control center of the cell, housing our DNA, the blueprint for all cellular processes. Insulin influences gene expression by interacting with transcription factors, which are proteins that bind to specific DNA sequences to regulate the transcription of DNA to messenger RNA. One key pathway through which insulin exerts its effects is the MAPK pathway, which not only helps regulate glucose metabolism, but also impacts cell growth, differentiation, and survival by altering gene expression. In healthy cells, insulin promotes the expression of genes that are involved in glucose uptake and lipid synthesis, enhancing energy storage and utilization. However, when insulin resistance occurs, these normal regulatory mechanisms are disrupted. The cells become less responsive to insulin signals, leading to altered expression of genes that can exacerbate the metabolic dysfunction. For example, in liver cells, insulin resistance can increase the expression of genes involved in gluconeogenesis, the production of glucose from non-carbohydrate sources, further raising blood sugar levels. Moreover, insulin resistance can activate inflammatory genes, leading to increased production of inflammatory cytokines within the cell. This inflammation can further impair insulin signaling, creating a feedback loop that exacerbates insulin resistance and leads to more severe cellular dysfunction. The alteration of gene expression due to insulin resistance not only affects metabolic functions, but can also lead to changes in cell growth and death, contributing to complications such as fibrosis, cancer, and cell aging. Therefore, understanding and managing the nuclear response to insulin is crucial for preventing the wide array of pathologies associated with insulin resistance. Therapeutic approaches aimed at improving insulin sensitivity often focus on modulating these nuclear effects. This can involve pharmacological agents designed to specifically target transcription factors and signaling pathways affected by insulin resistance, as well as lifestyle interventions that can indirectly influence gene expression through improved insulin sensitivity. By dissecting the nuclear responses to insulin resistance, we gain deeper insights into the cellular mechanisms that underpin this complex condition. The understanding is essential for developing more effective strategies to combat the widespread impacts of insulin resistance on human health.
Continuing our journey, welcome to part 9 of Inside the Cell, Unraveling Insulin Resistance. In this part, we explore the process of autophagy, a critical cellular mechanism for maintaining homeostasis and health by breaking down and recycling cellular components. This process plays a vital role in cellular cleanup and renewal, and it's intricately linked to insulin signaling. Autophagy, which literally means self-eating, is a cellular degradation pathway that removes damaged organelles, misfolded proteins, and pathogens. It helps maintain cellular function by clearing debris and recycling materials to support new cell growth and energy production. Insulin resistance has a complex relationship with autophagy. While insulin generally suppresses autophagy by activating the mTOR pathway, which promotes growth and protein synthesis, in a state of insulin resistance, this suppression can be disrupted. In healthy cells, the inhibition of autophagy by insulin is a normal response to the presence of sufficient nutrients. It prevents excessive breakdown and ensures that energy is directed towards growth and storage. However, when cells become insulin resistant, this regulatory balance is lost. The cell may not adequately respond to insulin signals to suppress autophagy when nutrients are abundant, leading to inappropriate or uncontrolled autophagy. This dysregulation can result in excessive degradation of essential cellular components, contributing to cell dysfunction and disease. Furthermore, chronic insulin resistance can impair the initiation and progression of autophagy, leading to the accumulation of dysfunctional proteins and organelles. This accumulation can exacerbate cellular stress and contribute to the development of diseases like type 2 diabetes, cardiovascular diseases, and neurodegenerative disorders. Strategies to enhance proper autophagy in the context of insulin resistance involve both pharmacological and lifestyle interventions. Exercise, for instance, is known to stimulate autophagy in a beneficial way, helping to clear out cellular debris and improve insulin sensitivity. Dietary approaches, such as calorie restriction or intermittent fasting, have also been shown to activate autophagy, offering potential therapeutic benefits by improving cellular health and function. So as we uncover the role of autophagy in maintaining cellular health and its interaction with insulin signaling, it becomes clear how essential this process is for combating insulin resistance and promoting longevity. Understanding and harnessing the power of autophagy can provide us with powerful tools to improve metabolic health and stave off disease. Welcome to part 10 of Inside the Cell, Unraveling Insulin Resistance, where we'll discuss actionable strategies and innovative treatments that can help prevent and potentially reverse insulin resistance, empowering you to enhance your metabolic health and reduce the risk of associated diseases. Understanding the multifaceted nature of insulin resistance, as explored in our series, underscores the importance of a comprehensive approach to management and prevention. Lifestyle changes are the cornerstone of any strategy aimed at improving insulin sensitivity. Regular physical activity is paramount. It not only increases glucose uptake by muscles, but also improves the efficiency of insulin signaling pathways. Aerobic exercises, strength training, and even moderate activities like brisk walking can have profound effects on insulin sensitivity. Diet also plays a critical role. A balanced intake of nutrients focusing on high fiber, low glycemic index foods, and healthy fats can help manage blood glucose levels and reduce insulin spikes. Diets rich in whole grains, lean proteins, and a variety of vegetables are recommended. Additionally, reducing processed foods and sugar intake is crucial for maintaining optimal insulin function. Sleep and stress management are equally important. Poor sleep quality and stress can exacerbate insulin resistance by influencing hormone levels, such as cortisol, which can interfere with insulin's effectiveness. Ensuring adequate sleep and employing stress reduction techniques like mindfulness or yoga can significantly benefit insulin sensitivity. For those already experiencing insulin resistance or at high risk, pharmacological interventions may be necessary. Metformin, for instance, is a commonly prescribed medication that improves insulin sensitivity and lowers glucose production in the liver. Newer drugs focus on targeting specific pathways involved in insulin signaling and glucose metabolism, offering more personalized treatment options. Emerging therapies, including those that harness the potential of gene editing or focus on molecular targets involved in insulin signaling and cellular metabolism, are also on the horizon. These treatments aim to correct the underlying cellular dysfunction that contributes to insulin resistance. Lastly, regular monitoring and consultation with healthcare professionals can help tailor these strategies to individual needs, ensuring that interventions are effective and sustainable. As we wrap up Part 10 and conclude this series, remember that the journey to improving metabolic health is continuous. By understanding the cellular mechanisms of insulin resistance and adopting holistic lifestyle changes, you can significantly enhance your body's insulin sensitivity and overall health. So as we conclude our in-depth exploration of inside the cell, unraveling insulin resistance, we reflect on the intricate dance between insulin and our cells, a relationship that is fundamental to our well-being. Throughout this series, we've journeyed through the cell, examining the multifaceted roles insulin plays in regulating blood glucose levels, maintaining energy balance, and supporting cellular functions. From the insulin receptor's signal transduction to the crucial work of mitochondria and energy production, each step is interconnected, and disruption in these processes contribute to the complex condition known as insulin resistance. We've seen how insulin resistance can impair glucose uptake, affect lipid metabolism, trigger inflammatory pathways, and alter nucleus gene regulation. Each of these factors does not just contribute to the risk of developing diabetes. They are also implicated in a broader spectrum of health issues, including cardiovascular diseases, obesity, and even neurological conditions. The knowledge we've gained underscores the critical importance of maintaining insulin sensitivity for our overall health and longevity. This series also highlighted the significant impact of lifestyle choices on our cellular health. Regular physical activity, a balanced and nutritious diet, adequate sleep, and effective stress management are not just good practices. They are essential strategies that can enhance our body's responsiveness to insulin. For those facing insulin resistance or even diabetes, these lifestyle modifications, coupled with medical interventions, can make a profound difference in managing and potentially reversing these conditions. As we move forward, let's carry the insights from inside the cell into our daily lives. Let us be mindful of the foods we eat, the physical activities we engage in, and the schedules we keep, knowing that each choice can influence our cellular health. Furthermore, emerging research and advancements in medicine offer hope, promising more precise treatments targeted directly at the mechanisms we've discussed. These treatments could one day transform the way we manage insulin resistance and metabolic health. Thank you for joining us on this cellular journey. We hope this series has not only informed you, but also inspired you to take proactive steps towards better health. The fight against insulin resistance is ongoing, and armed with knowledge, we are all better equipped to face it.
Thank you so much for watching. Please don't forget to like, subscribe, and comment on the video if you have any questions or if there's anything we can help you with.
Don't forget, Mindful Diabetes Incorporated is your resource, so please use it as much as you can. I'm Joe, and have a wonderful day.